Right. For this particular video today, we are going to be looking at something known as synapse or synapse. Now, some students have asked me the question, what is the correct pronunciation for this particular word? Is it synapse or is it synapse? Synapse is the British pronunciation and synapse is the American pronunciation. So for our Cambridge level, we actually use the British syllabus, so we have to go with synapse. But you know, when we answer the exam questions, it is not an oral examination, it is a written examination, so it doesn't matter. So if I use the word synapse or synapse, it is just the same thing, okay? And I tend to just switch between the two without realizing it. Now, before we talk about the synapse, see, I'm getting a bit self-conscious in the way I'm pronouncing it. Before we get into the synapse or synapse, we have to understand something very interesting here. Now, when I'm drawing out the sensory, relay, and motor neurons over here, as you can see, you notice something rather interesting. In reality, these neurons do not directly touch each other, okay? Now, some teachers may liken or compare our nervous system to an electrical circuit that you might have studied in science. Now, the electrical circuit, for it to function, you have the dry cells and you have the bulb over there, the menthol. And when the circuit is complete, which means the wires are all connected to each other, the circuit is then complete and the bulb is able to light up. And of course, if there's a tiny little gap, the circuit is incomplete and, you know, the bulb or the menthol will not be able to light up. So a lot of students assume that our nervous system functions the same way, that the nervous system, the neurons must directly be touching each other. But in reality, they're not, okay? There are slight gaps between the neurons, as you can see over there that I'm writing out, the word gap. So synapse, by definition, just means it is the junction or meeting point between the two neurons. Where I've highlighted in a bit of a purplish color, if you can see that, that is what the synapse is. The synapse is just the junction where the two neurons meet. Some students will say the synapse is just the gap. No, it's slight. It also has to include the neuron slightly before the gap and a little bit of the neuron slightly after the gap. And of course, what is the direction of the electrical impulse? The direction of the electrical impulse in this case is always moving from the sensory to relay neuron to the motor neuron. And if we were to just zoom in into the synapse area, any neuron that is before the gap is referred to as the presynaptic neuron, and any neuron that is after the gap is referred to as the postsynaptic neuron. So if we were to focus on the relay neuron part and the motor neuron part, okay, then in this case, the relay neuron becomes the presynaptic neuron because it's the neuron before the gap. Okay, as you can see the direction of the signal. So we refer to this as the presynaptic neuron for the relay neuron, and the motor neuron is referred to as the postsynaptic neuron. Now, some students will ask the question, so in the exam, am I supposed to use the word sensory, relay, or motor neuron, or am I supposed to use the word presynaptic neuron and postsynaptic neuron? Like what do I have to answer in the exam? It really depends on how the question is being asked. If they just give you a diagram of that particular neuron over there and they say, label what's the name of this neuron, then you have to say sensory neuron, you know, because you have to know the appearance of the neuron. But sometimes in the exam, what they will just do is they will zoom in to this particular part over here. And when they zoom into this particular part over here, you don't know what neuron is the one on the left and what neuron is the one on the right. The one on the left might be sensory, the one on the right might be relay, or the one on the left might be relay and the one on the right might be motor neuron. So you don't know for sure. But what you just have to say in this particular case, if they show you a structure of a synapse, you just have to know that when you see those ball-like structures, those ball-like structures will represent the presynaptic neuron, and those parts where you have those cups, which I'm just circling in red, okay, as you can see, a red circle, it looks like a cup, that is always representing the postsynaptic neuron. So What we're going to see over here then is we are then going to zoom into that one particular part. I'm putting a red square over there and I'm just going to zoom in on that. And we are going to see the structure further in detail. So you see the ball-like structure will represent part of the presynaptic neuron because it's before the gap, and the postsynaptic neuron is after the gap. So in all cases, the impulse is always going to move from the presynaptic neuron to the postsynaptic neuron. Okay, and that's another thing that we have to cover. Because they're not directly touching each other, when the impulse reaches the end of the presynaptic neuron, the impulse cannot jump. So one of the weird questions is, how does the electrical impulse move from the presynaptic neuron to the postsynaptic neuron? Because there's a wide gap. So we're going to look at that part later. For now, we have to first cover the structure of the presynaptic neuron. And in the previous videos, I told you that we actually looked at the end of the neuron, and I mentioned something known as the synaptic knob. And I said that the synaptic knob has these vesicles with neurotransmitters. And that is what we are going to be looking at right now in detail. So when you're talking about the presynaptic neuron over here, there are a few things that you have to cover in detail. Under the presynaptic neuron, I'm just drawing out these things. They are known as vesicles. Vesicles are tiny little vacuoles inside the cells. And these particular vesicles contain a very important chemical. The name of the chemical is known as neurotransmitters. You don't have to memorize the definition of neurotransmitters, but they are just chemical messengers that are released from neurons, okay, and they are used to stimulate another cell, usually another neuron or a muscle. We will talk about neurotransmitters in detail later, but examples of neurotransmitters may include things like dopamine, they may include things like serotonin, and one of the more favorite neurotransmitters for our syllabus is a neurotransmitter known as acetylcholine. So we're going to see acetylcholine in detail later. So neurotransmitters are just these chemicals that are just used to send signals. They are chemical messengers, by the way. And on the presynaptic neuron, there is a channel called the voltage-gated calcium ion channel. I'm going to put that in blue. And on the postsynaptic neuron, if you notice on the cell surface membrane, I'm drawing out sodium ion channels. Now you might be thinking, sodium ion channels, I've heard of this before. Okay, because we've covered them in the previous videos, right? But these sodium ion channels are not voltage-gated sodium ion channels. Because remember, voltage-gated means they will only open at specific voltages and close at specific voltages. However, these sodium ion channels are referred to as ligand-gated sodium ion channels, where they open when chemical messengers attach to receptors. In chapter 4 of AS, a long time ago, I told you that ligands are just another name for chemical messengers. So what chemical messenger do you think we are talking about here? We are probably talking about neurotransmitters in this case. Because if you notice the shape of if you notice on the surface of the sodium ion channel, I'm just circling a structure in green, that is a receptor. And that receptor, if you notice, is complementary to one of the neurotransmitters. They can bind to each other. So that means there is a connection. So when a neurotransmitter binds to the receptors, it causes the sodium ion channels to open. So this is a very specific type of sodium ion channel as well. So what we are looking at over here is a generalized structure of the synapse. Synapse. Okay, one or the other. Yeah, one or the other. Okay, both pronunciations are fine. Okay, but so if you get confused with synapse and synapse, just as a reminder, they mean the same thing. All right. A thing that I forgot to mention as well is, I'm just circling it in green over there. The gap between the presynaptic neuron and the postsynaptic neuron is referred to as the synaptic cleft. The word cleft just means a gap. Okay, so just be aware of that term as well. Now, There are many different types of synapses, but just to name a few, some of the synapses are referred to as cholinergic synapse, dopaminergic synapse, and adrenergic synapse. The reason why they all have different names is because each of them use different types of neurotransmitters. For example, the adrenergic synapse, you don't have to memorize this. It uses a neurotransmitter known as noradrenaline. Dopaminergic synapse uses dopamine. Some students will ask, is dopamine the feel-good neurotransmitter or the feel-good chemical? Yeah, it regulates pleasure feelings and such, okay? But I'm not a psychologist, so I'm not going to go into that. And the cholinergic synapse is the one that you have to know, because the cholinergic synapse uses a particular type of neurotransmitter known as acetylcholine. So you have to know acetylcholine, also referred, you can write it as capital A, capital C, small h. That's the short form. Now, so for your level, you have to know cholinergic synapse. And what's the meaning of a cholinergic synapse? They use the neurotransmitter known as acetylcholine. What does it mean by using a neurotransmitter known as acetylcholine? So let's look at that in detail. All right. The important question that we have to see over here is, how do impulses get transmitted along the synapse? So we have the cholinergic synapse here. I'm just relabeling and rearranging the wording so we can see it more. A thing that I want you to understand is the postsynaptic neuron right now. If you notice, I'm writing out charges over there. It's a little bit small, but I hope you can see the inside has a negative symbol and the outside has a positive symbol, which means to say in this particular situation here, the postsynaptic neuron is maintaining a resting membrane potential of about negative 70 millivolts. The problem is, as you can see, the presynaptic neuron is also maintaining a resting membrane potential. And I'm also just coloring it, by the way, so you can see it more clearly. One of my students have said that sometimes when I do it in a white background, everything becomes too bright. So I'm just trying to color the blue color parts. So here's another, I guess a nicer diagram, if you can see this, that shows you that the presynaptic neuron and the postsynaptic neuron are both right now maintaining a resting membrane potential. So the question is, how exactly does it send the impulse from one place to another? Because in reality, the impulse cannot jump from the presynaptic neuron to the postsynaptic neuron. Now you might be thinking, oh, but we had the myelin sheath. Yeah, but there's no myelin sheath connecting the two of them in this case. What we're going to see is we're going to break it down into 3 different pictures so you can actually see how it works. So the presynaptic neuron right now, as you can see, the axon over there, it undergoes a depolarization. So can the depolarization jump to the postsynaptic neuron? No, it cannot. When the impulse reaches the end of the presynaptic neuron, it changes the voltage in the area and it causes the voltage-gated calcium ion channels to open. Now, when the calcium ion channels open, calcium ions will rush into the axon. You might be thinking, wait a second, you've never mentioned anything about calcium ion channels and calcium ion concentration gradient before. Yeah, I didn't. In biology, sometimes in A levels, it's a bit annoying. They just introduce completely new things out of nowhere. Just write it like that, okay? Sometimes we just have to soldier on with, all this knowledge. So that's why it's a lot of things to study for this. So the voltage-gated calcium ion channels open and calcium ions rush into the presynaptic neuron. Now, when the calcium ions rush into the presynaptic neuron, it will stimulate the vesicles and the vesicles will start moving. Now, where does it move? The vesicles will start moving towards the cell surface membrane. And when they move towards the cell surface membrane, let's look at the second image, the vesicles fuse with the cell surface membrane and the neurotransmitter, which is acetylcholine, will be released. What's the name of this process, by the way? Chapter 4, this is exocytosis. Do not say exocytosis of the vesicles. No, it is the vesicles fuse with the cell surface membrane and exocytosis of acetylcholine happens. You have to be a bit careful. Some of my students tend to say exocytosis of vesicles, and that is wrong. The vesicle doesn't go out. The vesicle just becomes a part of the cell surface membrane. It is the acetylcholine that is released out by exocytosis. So, and the ACH diffuses across the synaptic cleft, which is the gap, that is number 5. And what does it do? The ACH will bind to complementary receptors on the postsynaptic neuron. And when they bind to the complementary receptors on the postsynaptic neuron, remember, those channels, those sodium ion channels are ligand gated channels, which means to say they only open when a chemical messenger attaches to it. And in this case, the chemical messenger being acetylcholine. So the sodium ion channels open. Remember, it was maintaining a resting membrane potential. Sodium ions rush in and the postsynaptic membrane depolarizes. And I told you before, okay, when one part of the membrane successfully depolarizes, guess what? The other part of the membrane depolarizes, which causes the other part of the membrane to depolarize, and therefore the postsynaptic membrane is now depolarized. That's how you are able to send the impulse from one neuron to the other neuron, even though they are not touching each other. Okay, so this is from number 1 all the way until number 7. So I hope you can see my writing. I know it's a bit small, so I do hope you can actually see that, because there's a lot of things I have to cram inside here. Anyway, all right, but let's try this again. Okay, if you want to see it all again in just one shot, okay, so the first thing that happens over here is the depolarization reaches the end of the presynaptic neuron. What happens then? It causes the voltage-gated calcium ion channels to open. Calcium ions rush into the presynaptic neuron. This causes the vesicles to move to the cell surface membrane, and it causes exocytosis of ACh. And the ACH or acetylcholine will diffuse across the synaptic cleft and bind to the receptors on the postsynaptic membrane, which causes the sodium ion channels to open. Bear with me, okay? I hope I've not lost you yet, okay? Take your time. If you have to rewind, go back and rewind. So number 6, the sodium ion channel open, the ligand gated sodium ion channels open, sodium ions rush in, which causes the postsynaptic membrane, or you can just say the postsynaptic neuron, to depolarize. That's how you generate the action potential on the postsynaptic neuron. That's it. I didn't say that's basically it, because whenever I say that's basically it, a lot of my students will just look at me like, really? That's basically it? Are you kidding me? Okay, so yeah, that's it. So yeah, it can be a lot to take in as well. Now, the last part that we also have to see is we have to see what happens to acetylcholine. Because if you remember, the acetylcholine was attached to the receptor, which causes the sodium ion channels to open. So this is to allow sodium ions to go in and cause depolarization. But if the acetylcholine is attached to the receptor all the time, then the channel always remains open and the membrane is permanently depolarized. And this is not good. We do not want the membrane to be permanently depolarized, because then it cannot return back to repolarization and resting membrane potential. So you want it to depolarize, repolarize, and return back to rest. And it cannot repolarize and return back to rest if the sodium ion channels remain open all the time. So what first needs to happen is the ACH or acetylcholine detaches from the receptor. And when it detaches from the receptor, guess what? That's a good thing. It causes the sodium ion channels to close and the membrane can, repolarize and do whatever it has to do. But we also don't want the ACH to be in the synaptic cleft, because if it's just located in the synaptic cleft, it can just continue to attach, detach to the receptors, and we don't want that to happen, because it will generate unnecessarily more action potentials that we do not want. So in that case, we have to remove the acetylcholine. Now, in the synaptic cleft, there is an enzyme, and the name of the enzyme is called acetylcholinesterase. Yep. Bear with me, okay? Just bear with me on this. I didn't come up with the name, okay? So if you are annoyed, you should not be annoyed with me, okay? I am merely a messenger, okay? So students, whenever I teach synapses to students, like, they have that, yeah, that look. That's the look they have, okay, right there. So it's just a lot to take in. So, so there is an enzyme called the acetylcholinesterase, and what it does is Oh, by the way, you have to memorize the name of that enzyme. Yeah, so what that enzyme does is it will break down ACH into two things, acetate and choline. And the choline is a chemical that is usually reabsorbed back into the presynaptic neuron, and you can use that to regenerate ACH. So for the most part, nothing goes to waste, okay? Not everything goes to waste usually. So you just have to know that the acetylcholinesterase breaks down ACH into acetate and choline. Choline is the more important one, and choline is reabsorbed into the presynaptic neuron and regenerated back to ACH. So let's do this all back together, summarize the whole thing. First thing first, depolarization reaches the end of the presynaptic neuron, calcium ion channels open, calcium ions rush in, vesicles move to the cell surface membrane, exocytosis of ACH happens, ACH diffuses across the synaptic cleft and binds to the receptors on the postsynaptic neuron. Ligand gated sodium ion channels open, sodium ions rush into the postsynaptic neuron, and the postsynaptic neuron depolarizes successfully. Then what happens? Acetylcholine detaches from the receptors, broken down by acetylcholinesterase. It forms choline and acetate, but the choline is reabsorbed back into the presynaptic neuron and regenerated back into ACH. Whew, that was a lot. All right, so this is going to be this you have to memorize, okay? But the good news is in most cases the question is quite straightforward for the exam. They would usually just ask, explain how the impulse is transmitted across a cholinergic synapse. So you just have to mention, and usually it's 5 marks. So any of the 5 marks, any points which are valid that I've just mentioned are good enough. I'll also put all the points in the description of the video when you're making your notes and you want to write it out back again. Okay, so I hope our synapses are all still fine. None of our synapses are fried after that particular lesson. So yeah, and we will stop this video here because it was quite a lot, and I will talk about the function of synapses and the importance of the synapse in the next video.